Okay, let's have a brief recap on protein substrate localization. Eukaryotic cells have organized themselves to put particular smaller compartments into them. They are typically lipid embedded, so there's a layer of membrane around it. That's the outer membrane. And one particular one here that you see is the mitochondria, of which we have several. Have you ever heard where eukaryote got the mitochondria from, for instance? And that's a little bit the idea with these compartments. But mitochondria is pretty much established. So essentially this is gobbled up from bacteria. So the mitochondria in a eukaryotic cell used to be essentially a bacterial cell that the early ancestor of the eukaryotic cell has gobbled up. It has completely rewounded, so mitochondria currently produce energy. And they cannot live alone they could not live alone as a bacterial cell. The mitochondrial genome is about the size of a typical, it's much smaller than that of a typical bacterium, and most of the proteins coded in the mitochondrial genome are actually not acting in the mitochondria. So the mitochondria is not that the genome is cut and whatever the mitochondria does today is in the mitochondria. This is not the arrangement. So most of what happens in mitochondria, we assume that there are about, in human, about 1,000 to 2,000 proteins acting, so like in a typical bacterium, but all of those come from the nuclear genome. So all of those are from the eukaryote. Anyway, and we're trying to predict substrate localization because that is one indication of function. Here are examples of larger constructs. That's the nucleosome that maintains DNA, the ribosome that does the translation. Collagen is just long fibers that give structural support. Those capture light. This is a molecular motor, and that motor in fact turns around here. And then there's signaling transport across G proteins. They allow transport of signals through the membrane, typically through the outer membrane. So that in fact is also the reason why subcellular localization is relevant, because most drug targets are actually on membrane proteins or on secreted proteins. In fact, at this point, about 60% are these signaling molecules, G-coupled receptors, so-called G-coupled receptors that I showed you before. So identifying sort of the secretory space, so everything that is secreted and is in the membrane, is relevant for a variety of concerns in drug design. Mislocalization, so if you have a mistake in, for instance, the localization signal in the signal peptide or in some other way that leads the protein not to act in the compartment in which is native, in which it's supposed to do its function, that typically leads to disease. The reason why not every single one leads to disease is because we essentially have a lot of backup proteins. We have high redundancy. That is one particular one where you see that the non-regular way of being localized, in fact, leads to a severe problem. And that again, so the knowledge is, why do we want to predict substrate localization? The knowledge is relevant for understanding something about function. It helps to identify may help to identify drug targets. And again, there are examples where disease states relate to different substrate localizations. So if I knew what is predicted and I saw what is observed, maybe from the difference I already could guess that something with this protein goes wrong. This is one example. More generically, because of all of these reasons, primarily because of reason one, the prediction can in fact help to design the next experiment. Since it helps me to have some idea, get some idea about function, it helps me to decide what in an experiment I could do in order to maintain, if I have some idea where it sits. The idea of subcellular localization continues to be that the transport through the cell, so where the protein ends up in the cell, is coded for by zip codes. Most of those zip codes we don't know. Some of these zip codes we talked about are simple in the sense they are learnable by machine learning because they're at the beginning of a protein or Some of them are at the end of the protein. We did not talk about that because they don't work very well. We talked about the NLS, where the NLS motif, the zip code, so to speak, is somehow spread all over the protein. Now, how could you imagine that something that is spread anywhere in the protein could be a zip code? Can it be anywhere in the protein? So imagine the protein is a sort of extended sphere. This is a totally simplified assumption, but it's not such a bad model. In a simplified sort of extended sphere model, can the zip code be anywhere? So say there are 400 residues in this protein. Can it be any of those 400 residues?
I would guess you need to distinguish between surface.
It has to be on the surface. So the entire idea of a zip code is just like in the letter where the postman sees it. Here somebody has to recognize the zip code. And yes, it can be anywhere, so you cannot necessarily predict it has to be on residue 420, but it has to be on the surface. So that much is clear, right? Now, there are high throughput methods to, in fact, see or experimentally show subcellular localization. Mostly this is what people call tagging with GFP. So you put a report, what the biologists call a reporter protein. GFP is green fluorescent protein that when you shine light on it, it is fluorescent green. And essentially all you do is you put this GFP, which is an entire protein, you clone this in the genome to be next to the protein that you want to see. Okay? It is sort of co-expressed with it. So essentially you have the regular, let's call this the GFP, you have the regular protein, and you put the GFP next to the protein. Those two things are expressed. This does its regular thing, and GFP shines light or reflects light, is fluorescent. And that's the way you really, so that here you see in fact 3 fluorescent proteins, the red, green and blue, and that allows you to really get an idea where every single protein is. If you attach the GFP to every single protein out there. You cannot quite do every single protein, but really biologists do thousands of these. So in the bacterial genome, they take thousands of proteins, and to every single one, they engineer a way of the genome where this GFP is next to it. And then they shine light. Again, in this entire bacterium, or in yeast, as a typical, this was where it was first started. Yeast has 6,000 proteins. So you essentially need 6,000 different cells. So in every one there's a GFP to another, attached to another protein, right? That's the way it works. Now, the problem with that is, well, in some cases you see there's something in the envelope, you see it's outside, you see this sort of nuclear, but there are possibly mistakes. And this is a high throughput method. You can do a lot of proteins with this, but you're not extremely accurate. And it's still not complete, so you can in fact not do that for 6,000 proteins in yeast because of experimental issues. So this is a slide from Tatiana Goldberg. For yeast, which in fact has about 6,600 proteins as is shown here, only 5,000 have been annotated. I'm saying only because this is 15 years of people trying to get 6,200 or 6,600, right? Which is 5,000 is close, very, very close to 6,000, but it took a long, long time. And so there were generations of Nature papers who claimed that we have now the complete yeast with this high throughput. It was still not there. But nevertheless, it's fairly close, but there are some issues with the accuracy. Again, I talked about the sort of hierarchy of the sorting mechanism for the transport in the cell. There's one major path to becoming secreted, and these transports are done differently. I talked about different types of signals. One that is sort of recognizable on the linear sequence, one that is only recognizable once the protein has folded up. This is the kind of thing that Ole said, it has to be on the surface. So 2 different regions in sequence. Two different pieces come together, but they are on the same surface. This is why the zip code can be recognized once it's folded. The idea of zip code goes back to Günter Blobel, who got the Nobel Prize for it. Okay, There are more Nobel Prizes on this issue of zip codes. This is a set of more recent prizes in 2013. And we talked about the prediction by sorting signals, homology-based, text-based analysis, and de novo, and then sort of hybrid approaches that use a little bit of these. In fact, I will go more into the next particular example of that today. Now, whenever you predict in this field, the data is noisy, that's an issue here. Prediction performance must be established properly. We talked about this three-way of training, cross-training, testing. We talked about establishing of scientific significance and statistical significance. We talked about being very careful in the way you filter your data sets, for instance, to exclude sequence redundancy. Sequence redundancy means that you do essentially the same experiment for very, very related organisms, where you assume that you can apply homology-based inference, and that the method that you typically develop with machine learning is not relevant for that. And this is another issue here that, again, we have a lot of experience in this field because computational biology is really between two fields, but you will have a lot of that in the future, no matter what you do. Most likely, at some point, you will meet machine learning. Machine learning is one of those things, I believe, that is not only hype, but it will be with us and it will change the world. Maybe other than other hypes. Many hypes are hype for a few years. This will stay with us. I'm convinced about that. And it is already changing the way we do computation. So it will be with you. And there will always be this issue, how do I present? It's not only how do I present the results to the user. This is not only about interfaces, human-computer interfaces. This is not only about how to visualize. This is part of it, but it's much more than that. You, as a developer of the tool, compute performance measures, some area under the curve. That is sometimes relevant, but sometimes not. So you have to find out what is relevant for the user. They cannot tell you. So they are typically, so we see that in particular in interaction with clinicians. Clinicians are happy when we can predict something because they're random on some of the data that they have. They really have random guesses. And so if we do anything, that does help them and they're happy. But then the area under the curve is not relevant for them, because ultimately the area under the curve is a statistical measure for thousands of people. In front of them is one patient today, right? So it's a very different focus. And we have to learn, and they have to learn what they want, and we have to learn what they want. And it's a process. So this is highly tricky. And it's part of machine learning, and it's part of a big challenge in it. The benchmarking of prediction methods is very, very complicated. Again, machine learning in most applications today is such that you're not benchmarking because the tool will not survive very long, or you're essentially doing one thing that is relevant only for today. In the kinds of things that we do, where you develop a method that then sits on the web sometimes for a decade or longer. So some of my code is around since 27 years, and it's still running, and it's still being used by people, which by the way is horrible if that ever happens to you. It's the worst thing that can happen to you. Anyway, so this benchmarking issue typically is taking more time than everything else in machine learning. For most people it's the other way around, but if you do it right, it takes a long time. Okay, now let's get back to, or not back, fast forward to a new idea of doing the prediction. And that new idea is supposed to, so far we really focused on eukaryotes. Now the method from Tatjana here should somehow mirror this tree through some sort of decision tree, the type of thing that I introduced before. But it should also predict for eukaryotes, bacteria, and archaebacteria. And in fact, we'd like to have sort of one basic prediction framework. We want this framework to be robust against sequencing mistakes. So this is one issue that we have not discussed so far. We have always talked, I've presented to you this idea that you take the amino acid sequence of the entire protein. You translate it into what fraction of alanine is in this protein, and that's one input to this, for instance, 20 input vector of all the amino acid composition states, right? But the moment somebody makes a sequencing mistake, okay, if you get one amino acid wrong, and the protein is 400 long, this vector will not change much. That's absolutely clear. But imagine somebody cuts out 30 residues. and cut out 30 residues is something that actually happens very frequently. I have briefly mentioned this. It's not really crucial that you remember this. Typically, there's say we call this whole thing here a gene. And I do not have a black pen, that is true. But I deliberately do it here just to highlight that it's not crucial for you to remember and have a pen. So don't worry that you don't see it very well. Now, these 4 things here, in a gene, you have regions. You have 2 types of regions. One region is called the exon. In this particular case, it would be exon 1, E2, E3, and E4. And then there are these introns. Introns are what is called spliced out. They're cut out. Okay? So introns are part of the gene, but part of the gene that will not be put into the protein. Only the exons, only those E3, 4 E's there, they will sort of be put together and they become the protein. Now sometimes in genomes, the way genome sequencing is done, maybe an exon is missed. So a protein with 400 residues may have 4 exons. And if you miss one, you immediately see. So you're not only cutting out 30 residues, you're cutting out 100 residues. And you're cutting out these 100 residues somewhere in the middle of it. So this will clearly influence this vector. The 20-dimensional vector, input vector of amino acid composition. Cut out the E3, it does not have to, but it can really change, right? In particular, if it is a longer thing, in particular, remember that domains are sort of 60, can be around 60 residues long. So what you could cut out could be an entire domain. And that domain may really be different from the rest. Okay? So that's another thing that we try to address this time. And again, we try to expand the model by doing a prediction for many classes. What I showed you so far, this log3 version, I can't quite remember how many states it had, I believe 7 or 8. But we want to expand this. So we have one for eukaryotes to have really 18 different states. We want to include membrane prediction in this. Again, we want to have this decision tree that resembles the cellular sorting. We want to have some sort of reliability. So we want to be able to say, if I say this is a nuclear protein, and that is a nuclear protein, and I want to distinguish between predictions that are strong and predictions that are weak. In both cases, I predict a nuclear protein, but I want to have some feel for what's the probability of predicting that it's nuclear. And I, of course, want to have a method that works very well. So now, the way she did it, and it's a little bit topsy-turvy here. Let's begin with a data set. So the data set, you have seen that already. We take essentially SwissProt, because in SwissProt we have experimental annotations of subcellular localization. And these are the numbers here in 3 classes for archaea, 6 for bacteria, 18 that Tatjana had in 2000. Oh, I can't remember, 11 maybe? 11 or 12. So this is 59 proteins, which is a very small number, as you understand, for 3 classes. So essentially you have 20 per class, 479 and 1682 for eukaryotes. That is the sequence unique version. This is just, I'm sorry for the quality. The original SwissProt file here that has an annotation in nucleus membrane, and so this is one of the membrane that will be in the state nuclear membrane. We will exclude proteins with non-experimental annotations and with unclear multiple localizations. Again, proteins travel between subcellular localizations. So if you have experimental information, it may very well be that you have, for the same protein, the protein is cytosolic and nucleus, because it shuttles between these two. We argue that there is always a dominant class. Dominant class means maybe the one that is for which the sequence is most optimized. Maybe the one that the protein spends most of the time in. Maybe the one that is most important for doing its function. And this argument is a little bit more complicated. I could discuss that offline. But if we stick to experiments that have only one localization, we may have some information there from incomplete experiments, but we bias our selection toward experiments that do not do high throughput. Because in high throughput you have the probability that you really see different things. And you will report different things. because your high throughput experiments will report whatever they see. There's no way to filter the data and say whatever I see strongest. Well, in some sense for the fluorescent data you can take the strongest light emission. But typically this is really done by people. They see light, they don't. And then they see light in 5 different places, so they annotate 5 different localizations. So typically this is also a way to select more careful experiments where one person really looks at what is the dominant thing, what is the most important aspect. And again, we want to sort of identify transmembrane proteins in this. In terms of homology-based inference, we know there's this HSP curve here, number of proteins aligned, pairwise sequence identity. Above the curve is the H-val 0, and you go further up, the blue points here represent pairs of proteins that have the same subset localization. The red ones, pairs of proteins that do have different subset localization. You see there are red points up here. So where you have a lot of residues aligned, a high level of sequence identities, that's very much above the gray line. But many points above the gray, if you stay high enough above the curve, say the curve plus 10, then most of what you see is blue. Most of it has the same subset localization. And that means that you can do homology-based inference. So when you see 2 proteins, for one you know the subset localization, for the other you don't. They are substantially above this curve. Then you have a high probability, a high chance to infer that the other one, for which you do not know the subset localization, is also the same. It's the same as the one you know it for, right? Homology-based inference. That means that when we apply this method, we want to be able to work on proteins for which homology-based inference does not work. That means we have to cut out a non-sequence non-redundant data set. And this is how the numbers suddenly melt, so to speak. So we start with 3,500 eukaryotes here, for which we know the subset localization experimentally very well. Then we apply this H-val of 0, and we are at a level that is much, much, much. I said 3,500? No, 35,000. So then we come to a level that is much lower. That's the data set I showed you before. Then for the data set we do what sometimes is called a stratified k-fold cross-validation. Actually then we did this 3-fold rotation that I talked about. So what it means is you take a training and a cross-training out, and you make sure that there's no overlap between training and cross-training. Yes, we established that already by taking out a non-redundant set just for security. You cross-check everything again. You pull in this case, since we have ended up in a sequence where we need a unique set with only roughly 1,600 proteins, that was not enough. So we had to pull in homologs again. That's what I briefly talked about on Tuesday. So the slide is not very good. Remember, this is the story that I talked about, where the experimental data for everything from SwissProt is the set E, and the sequence unique subset here is the small e in there, right? And now for training, what this one here signifies is for the training set, I pull in some from here. So I pull in homologs to train. And I make sure that whatever I pull in here, and that's the idea here, remains different from the test set. And in the test set, I still take the same that I had before, the small e. I only test on sequence unique data set, but I pull in some homologs only for training, for the training set here, up here. And those homologs pulled in for the training set, again, for every single one I pull in, I must make sure there's no overlap with the test set. This is just the way these clustering methods work. It increases the size of the data set by a factor of 4. So from 1,600 to, yeah?
No overlap in the test set and also in the training, right? No?
Say that again?
The overlap should be like, you should not have...
Oh, with the yes, only not with the test set. Because they will, by design, they have an overlap with the training set. So I'm pulling in things that overlap with the training set. Because in my training set, so I had this 1,600. Just let's assume we do a 4-fold split. And I have a cross-training set cross-training of 400, say a training of 800, and a testing of 400, right? Those are the numbers. Now, this 800 for training, they are not enough. So, for the 800 set, I try to pull in homologs. The homologs I pull in from this large E. So there are things that are sequence similar, that are above this. If I went back to this curve here, again, we do not quite take this curve, we do take the curve here plus 10, but assume we took the curve. Then my small E set is such that everything, every pair in the small E set is below this curve, okay? And that is what I will use also for testing. And my 800 there are also such that every pair in the 800 is below the curve. But now what I do is I pull in some from above the curve for the 800 to increase it. And quadrupling it, so I pull in essentially, so I essentially end up with 3,200 for training instead of 800. Now the 3,200 minus 800, so the 2,400 that I add in this. These 2,400, again, for every single time I add one, I have to make sure that it's clearly not above the curve with respect to any testing. I possibly could also do that with the cross-training, but we ignored that. We just want to make sure that when we establish the final data that in the testing, we are really sure. Is that clear, everybody? Is that? Yeah? Okay. Oops, that was too fast. So that brings us to the method now. That's my data set. I wanted to use an SVM, and in this particular case, so again, you know, the SVM finds the best way to distinguish the closest points that belong to either of the two classes. So we have class L and class non-L, or class L and class non-L, and the SVM will essentially put a support vector that defines this line to the points that are closest to it and separates them the best. It's a linear separation in the hyperplane, and we essentially do that by projecting a problem of this type into a problem where we can separate by simply going into hyperspace. And that's the kernel function. Now, the kernel function there are different types of kernels. There's simply a string kernel, there's a k-mer, so you match a k-mer. There's a mismatch kernel where you essentially have a certain number in the k-mer where you can say, okay, I allow a mismatch. It's very, very simple. And there is something that is called the profile kernel. Has been used for protein classification before. And this idea now is, okay, when I look at a k-mer of residues, let's not look at just any k-mer. When I look at a protein alignment, so I take my protein, okay, in this particular case I should put an alignment in here to make this point clear. So I have my query protein that is aligned to many other proteins. And maybe here is a position where they all have an H. And maybe here's a position where this one has an A, this one has an A, well, this one doesn't have any, this one has a C, C, G, or something. Now, what I see is that the point, this H here, is more important than this A. The A in that family, because I assume that every member of this family essentially has a similar function, has a similar subcellular localization. So I assume that having an A here is less important, because you see proteins can do the same subcellular localization with a different residue there. Maybe what I see is that, in fact, it is important not necessarily to have an A, but you need either an A or a C or a G. But in the other one, you really need an H. And you begin to see that here is a different type of k-mer that evolves from this. It's not the k-mer necessarily where you have H, E. Is there anybody? I have the bad feeling that HEPA means something. Does anybody associate anything with the word HEPA? in any language that any of you speak? Okay. So the word, the k-mer in this particular case, the 4k equals 4, the k-mer of HEPA is not necessarily what I want. So in this particular case, I may say the H is important, the E I will see, and the A I sort of put a probability k-mer type of thing. So I will use, say in this particular case, all three of those for my argument. HEP would be conserved throughout this family, and the A not. Then essentially I would do 3 k-mers here. I would do HEPA, now the way of pronouncing it gets complicated, HPC and HEP and G. So those 3 k-mers I would sample. Okay? And then I would simply find a way to count how often they occur. And this is exactly what this slide shows. So in this slide, the sequence runs in this direction. So that's the first residue, the second, and so forth. And those 20 here are the possible amino acids. Okay? And the entry simply says, how often do you see in this particular column? So I inverted in that presentation, the sequence runs in this direction, and this one here runs in this direction. And I replace here, down means essentially everyone is a protein. Here it's simply the 20 amino acids is a frequency. So I simply count, in this particular case here, for this column I would simply count 30% A's, 30% C's, and 30% G's. And here I would count 100% H, 100% E, 100% P. And that's the entry to this table here. Okay, then I apply some log. Again, the query up there, this is exactly the way I show it. And then I count. I could have actually done it on this example. So then I transfer minus log of this. And that leads to a situation where essentially have the following type of k-mer count. I present my protein as a vector for all possible k-mers in this conservation space. All possible k-mers are simply, if k is 3, is 20 by the power of 3. So 20 by the power of k, yes?
So k-mer is a substring, a common substring?
Yes.
Okay.
And the way we what we actually want to get to is to have a measure in the k-mer how frequent that is in that family. Not only in that sequence, but in that family. Okay, how conserved that is in the family. And the way we are going to do it is we are going to have some threshold. We're going to say, well, the HEPA, these 3 here are above a threshold because you see them more than some number of times. You see them often enough to say they are sort of a conserved feature. Okay? And essentially in this vector, we are going to have an entry if we have seen that k-mer here is played on 3 mers, 20 by the power of 3. So the vector has an extension of dimension of 20 by the power of 3. So 8,000. So essentially, again, in this alignment, we ask how often is a particular 3-mer, trimer, AAT, CAT, CT, Q, core conserved. Conserved, again, is defined by some threshold, which we have to then optimize. We have to optimize at some point the K, we have to optimize the threshold of conservation. But then the rest of the process is we are simply counting through. In this particular case, the graph here only shows the CAT. For the CAT, no, it shows which one. Which one do we see whether it sums? The CAT sums to, so if it is smaller than 5 or larger than 5, so the vector, I thought there's an animation, the vector switches now. So this one goes out, the smaller one than 5 ones go in. So essentially, as far as we go in the sequence here, there's 1, 1, 1, and everything else is 0 of the 8,000 components of this vector. And then you run through the sequence. When you see the same thing again, you just up the count. So this is not, the max is not 1 here in this table. You just add as long as you find another k-mer of this type. For the comparison of two proteins, you essentially do a vector product of these 8,000 dimensional vectors, right? And with this, you establish a similarity in this k-mer space between two proteins. Okay, are you seeing where this goes? Now, all I need to do in the next step is somehow figure out which of these are relevant k-mers, and in fact have one of those be the database, and to train my system and find the support vector that essentially define a particular class. So I'm going to have a classification of nuclear versus not nuclear, and the second protein then is all going to be my data set of nuclear proteins or my data set of non-nuclear proteins, and that is in fact where I try to find the support vector to distinguish exactly between these two classes in this 8,000 dimensional spaces for k equals 3. Now for k going up, this unfortunately scales fairly rapidly, so you go very high. The k is not written here. So ultimately here we compare it to different kernels. We essentially, so for Archaea the data set is rather small, the performance is very well, so it's unclear to see a difference between the kernels. But for eukaryotes we clearly see that this profile kernel works very well, so we can choose this profile kernel. Now the next issue here is how do we split our tree? So remember we want to build this tree where every node in the tree is one subset localization against all others. So it's nuclear or not. And so we could try an all against all approach. We in fact initially tried at this point in the project, we tried many things, trained many things, and tried different classifications, and ended up with something that is in fact close to what I showed you before, with a slight twist in this particular case. Previously the top decision was, is it going to be on the secretory pathway or not? Now the top decision is it's going to be in the membrane or not. And then for the, I can't remember which one is the membrane path. How would I see that? This is the membrane path here on the right. So for the membrane path, some of these decisions in between are similar to the non-membrane path. But every node here is trained on a different data set. Okay? So everything right here goes into the membrane path. So this will only see membrane proteins, or at least proteins that are predicted to be in membrane. Okay? So even if there is a decision that is similar, here is one where we go into ER or ultimately extracellular or Golgi. So at this particular split here, that is a similar split, except for the next states are different, but essentially this split here is ER or not, this split here is ER or not. It's the same type of split, but this will only see membrane proteins, and this will only see non-membrane proteins. Again, it will not only see membrane proteins because some are incorrectly predicted. We'll also see others, but in principle that's the idea behind it. For Archaea it's much simpler. Essentially you have cytoplasm, there's extracellular, and there's the membrane. For eukaryote, and those are the three states. For prokaryote you do have different states. There's the cytosol, extracellular space. This one is not an animation. And then you see the full tree here of 18 different states for eukaryotes. 18 different states is only for plants, in fact, because this is chloroplast, it's also chloroplast. So 2 states do not exist for non-plants. Okay, again, I said that. The first decision in this tree is, are you soluble or non-membrane? Right? Everything that is membrane is in red here. So on the red side, I should have waited for that slide. And the left side, essentially the red part is the... So this is about user face design or something like that. So what does this signify for you? What would you have said? Is this showing membrane or is this showing non-membrane? I mean, maybe it's too late because you now, some of you may wake up now, and so those are the ones I asked the question to.
I think it really depends if you showed us the picture earlier with what is important.
Dude, so you don't count because you didn't wake up only later. So is there anybody now? It's difficult to ask that question now. I understand that there is a motivation not to say I slept before, but I really I was too slow. I have to try that again next year. Okay, so I feel it's a strange choice but anyway, so Tatiana says everything red is essentially the state I'm looking for, okay? And then we go further, and that is essentially not copied from before, it's the best solution here. It is like before, so one goes into the secretary pathway here outside, and one goes into the not secretary pathway. And here are the performance estimates average over 5 different training sets. I believe we should just zip forward. Then there is a parameter optimization. Let's just go into the testing. So there's an accuracy. Again, this is often observed as precision. There are so many words and I always find that confusing. So Wikipedia has a bunch of definitions. People use a bunch of ways of calling it. I prefer the word accuracy for positives and negatives. Simply, so for some people there's a word for precision, it's only defining for the positives. I feel that it's easier to learn two words and then sort of keep in mind that there's a symmetry of the states. Whatever. So this is the way I do it. So there's an accuracy essentially of a state, and the accuracy essentially says how many of the ones that you predict at some threshold are right. Okay? In state P or N, so in the state, if you do nuclear against other proteins in the state nucleus or in the state non-nucleus. So there are two different numbers. Okay? Coverage is the sort of inverse in some sense. That is what sometimes, at least for the positives, is called specificity. Coverage is this idea, how many of the ones that I experimentally know are nuclear did I find? So if somebody is out there to find all secreted proteins in a bacterium, because those attack eukaryotic cells, then they want a very high coverage. They want to find as many as possible, because one of those may be the one that causes the disease I see now. Okay? Again, so this is of the ones that at some threshold you predict, the right ones divided by the ones that you got right, and the ones that you missed as secreted, as positives. Again, the state again comes in positives and negatives. So there's a coverage for the positives and a coverage for negatives. So that gets to the point that we already had. We actually did not discuss it. So this is a common situation that I find. So we have one method here, and it reports in this particular case, what I mean is a three-state performance. Three-state performance means I have to distinguish between proteins that are cytosolic, nuclear, and extracellular. And I ask for all the proteins that I classify in my data set, how many are right in either of those three states. That's what Q3 essentially means, a quotient of right, or in the percentage here. And now you see in a publication or in a thesis, you see these two numbers. And you infer that N1 is better than Web2, because the number is clearly higher here than that. Is there anything else that you would say if that would be the person you work with in the same room? If that would be your friend who writes that in a thesis? And typically then your friend developed method N, most likely. So you would most likely say, great, pat on the shoulder. That you should really do that. Very, very important. We need that. We need positive feedback. We all don't get enough of it. But there's a but in here. Who gives me the but? Yeah?
The difference is not that high.
This is a story that we have been discussing, and we have to clearly open that can. Before we open that can, there is something else. It relates to that, but it's, yeah?
You're comparing values to different significant places?
Significant places. This is an interesting word. So the word significant places is essentially exactly what you have to think about. So your friend most likely is happy that he or she has the better method. And being happy, since they also have the computer, they can put more digits. This suggests even more performance accuracy. And that is totally misleading. Okay, the word significant digit already is sort of capturing it. At least we should have the same number of digits, okay, in a table reported. But the next question, of course, the important question that if you had this one here, it is sort of written in the word significant in some sense. How many digits do you actually want to show? And how do you find that out? How many digits you want to show? I mean, often it's decided because you fit many things on your table and you just cannot show that many digits, so you cramp it. This is not the kind of decision. It does happen, it has also happened in paper, we have done that. Okay, but this is not what I mean here. What's the right choice? What's the right procedure?
I think in general you take the amount of digits you have for your input data, so basically the lowest kind of precision, but that might be hard in this one.
What do you mean by input? That's a very interesting idea. So you essentially say, I read in what you mean, what you say. I read you to mean Johannes, right? That you somehow look up the experimental error, and you sort of gauge the error that you get here. And this is exactly what I sort of meant on Tuesday about scientific significance. This is the kind of rationale that gets you from statistical significance to scientific significance. On this level here, the moment you decide the digits, first you should at least look at the statistical significance. So statistical significance brings us back to estimate statistical error. That's essentially what you said. And I mean, pressure. Presta. Ah, that's he is there. Presta. I'm sorry, it will take a while. Presta. So essentially the idea really, you have a distribution here. Say for some data set the average is 72. And that means here 72, that's the average, but then there's a distribution. Whatever I show, the feature is not relevant here. And the degree to which this is how narrow the distribution is the standard deviation. And the standard deviation, in order to get the standard error, you essentially divide the standard deviation by the square root of the number of data points that you have, and that gives you a rough estimate of the standard error. This is the statement here. And in this particular case, say the standard error is plus minus 0.8. So how many digits can I show? Should I show? How many digits are supported by the standard error? Okay, let's begin. Is this digit supported?
No.
Does anybody feel this digit is supported? Does anybody feel this is supported? This? Yes? Was there a yes? No. This one?
You can argue.
So this one is the argument. Okay, so this one you can argue about, right? Possibly not. Depends on how you look at the story. I would argue one standard error plus minus 0.8. One standard error is 1.6, so I would argue that this is actually an integer story. But this is, you can really argue about that. And if I cannot argue with you. But if you put more, now here comes the real story. This has happened in every single thesis that I've ever sat on. And not only here. At Columbia University, in other countries where I've done thesis, is almost in every single paper. I have seen examples where papers are rejected only because of that. Because with a simple argument But somebody who does not even know to estimate their error, how can I trust them to do the right science? Okay? There's so much in your paper that I cannot look into. Most of the time I cannot look into your code as a reviewer. This is something I can look at. And if you even don't get that right, poof. By the way, this brings us to a different point that I also have seen. On a very, very substantial level. I've seen grants being rejected because of typos with the same argument. You're a scientist. If you cannot even check mistakes in your procedure, in your writing, how can I trust that you check mistakes in your procedure? So be careful about these things. Some people can make you trip. In this particular case, I would argue this is not nonsense. This is really, really important to know. And if you don't think about estimating your error, then you truly made a procedural or methodological mistake. Okay? Okay, so maybe at the end of the day we end up with these two numbers. And that brings us to this issue of what else to consider. I talked about that already on Tuesday. You clearly have to consider sort of what is random. And there are different models for what is random. What is random could be just, you throw a coin. You could throw a coin, however, according to the population that you have. So if you had, very often we have in our work, two states that are very differently populated. For instance, we want to predict nuclear proteins against all other. Then the state of nuclear proteins in training will always be much smaller. Because I predict against all other proteins. So nuclear will maybe be 20% of all the proteins, right? So that means that the random number is not 50% for this. In the most extreme case, if we just look at a two-state performance, the random probability then is 80%. You always predict the majority state, right? This is nonsense, clearly. But you have to keep this in mind when you put the random. So at least you should define a random that sort of predicts according to the sample size. And then there is sort of a step in between here. Maybe there is a method that is totally dumb. So you have a great idea of doing a method. And a method that is totally dumb, as a composition. So I've talked about that. It's around since 30 years. You find it everywhere. So you should at least beat that, right? So you possibly may want to have some points in between here, where you, with your methods, just, let's call it, recode the bottom that you would absolutely want to rule out or make sure that you do better. This is not necessarily a way to establish that you're doing good, but it's sort of the minimal ones. And then, again, Johannes talked about it, we talked about it last year on Tuesday. We have to somehow establish what's the experimental error, because that's the ceiling. I cannot do better than that. If anybody claims that they have a prediction method, if the experimental error works, it'll be 95. And anybody claimed, I have a method that is 98% right. That is wrong. Okay? This is another thing. It's like not estimating the error right. It cannot be. There's something totally wrong. So at least most likely this is because the error estimate has been forgotten, or the error estimate was too small. So if somebody says, okay, I predict 98% with a one standard error of plus minus 5, then we're on the green, right? That means that essentially we're talking about 90% of the probability that you're within, for 98, that you're between 93 and 100. It's still high, but well, it's still high. It's still higher. Okay, but anyway, you get the idea. So, in this particular case here, the question was which method is best, and the idea here is there is a new method. So I don't like my slide because I introduce a new digit. So typically somebody, but again, this is, I've seen this before. So there was the new method that somebody had, and of course then written on top. And the new method was claimed to be better simply because numerically this is higher. And maybe somebody, so they, oh, here's another issue. Maybe I get into the slides later, but say you have your data set and you establish for your method 73.5. And the reason why you take 73 on this method and 1 is because you don't have access to N1. But you find the number 73 in a publication from N1. Okay? Can you compare these two numbers? Why not? Fair. Okay. So Web 2 and N1 have not been developed at the same time. At some point, or maybe they have been developed parallel. And I now write a review. So the developers of N1 put N1 at this number here. The developers of Web 2 put this number. I put it all together, writing a review, and I put these two numbers in a table. They have been developed, published roughly in the same month, let's say, okay? Is that okay? Can I do that as a reviewer? So in this particular case, they have the same number of digits. So I'm just picking two examples this is not the point I wanted at this point. I'm aiming at something else. So I'm just reconstructing the example such, yes?
Do they have the same data?
That's exactly the issue. We must, before we can do that, we must make sure that they use exactly the same test set and training set. Well, training set not necessarily, but as long as we trust that they did the proper cross-validation, what really counts is that they have the same test set. And that is the crucial issue here. That typically, that is very, very difficult to maintain. Now, let's look at this particular example. How would we rank these methods? What is my next slide? I'm not entirely sure. That's the issue, yes. So assume you have a table like that and you want to rank. So the first thing is you have to get to the error estimate. If you had data like this, here secondary structure prediction where you have an average of 72 and you have a distribution, it's very easy to get a standard error. I explained how. But if you have subset localization prediction, you have either the right prediction or the wrong prediction, and this you can only turn into an error estimate by sort of measuring the standard error through bootstrapping. And the bootstrapping idea really, some of you may know this character Münchhausen here, who pulls himself and his horse out of the sump on his hair, conveniently. And in some sense, so the method comes from Bradley Efron, in some sense is really like that. You use the data that does not allow you to get an error and get an error estimate by essentially sort of doing the take out of the set and creating new data sets, distribution of data sets by sort of taking your data set and taking, for instance, only half of it out, compute the average, take half out, compute the average, and do that 1,000 times, and you get a distribution. That overestimates or underestimates your error, but at least provides an error estimate for binary predictions. And then gets to the standard deviation. And I did not pull this example through right. Now, say in this particular case, the standard error would be 0.6 or 0.5, plus minus 0.5. How would you rank the methods? So if I asked you to name method rank first, can you name? I should have said methods.
So it's 0.5 for all models.
Yes.
I would say that A and B are the top ones.
So that's the crucial point I wanted to get at. Ole says there is no method that is top. There are two methods that are top. So let's begin there. Sorry for interrupting you, Ole. And since I cannot distinguish between these two, so this is numerically the highest, but I cannot distinguish it within one standard error to method A. So I call method A and B performing equal, they tie on rank 1. And then what? How does it continue? Can somebody else take over? So we have rank 1. Let's just write it down. One is A and B. Rank 2. And that's it. So again, I was about to write here. I shouldn't. The standard error is plus minus 0.5.
Could it be also B still? Because the standard errors, well, overlap.
So this is just edgy.
Yeah.
So now the problem begins. If it could be B, well, now you have the alternative that you say you double list methods. So B is both first and second. Or you say an alternative process or an alternative decision, in some sense equally valid, would be, okay, since you cannot distinguish between B and C.
But then again, if you have enough methods, you cover the whole space.
That's the problem with that.
Okay.
But there's still no way around it. So the point is, since you can argue that way, method A and method B may really have an incentive to stand up and say we are best, and numerically they are the highest. But still, this argument pulling through makes it very complicated. One way you can do it is you say, okay, we are ranking our methods A, B, C, D, E. And we essentially look in pairwise comparisons, and all the pairwise comparisons, how often is A significantly better than the others? And then we get a new ranking of that. And we essentially rank number one, the method that is most often statistically significantly better than others. And this in particular case would lead to the initial assumption of A and B being 1. That would separate out B and C, just because both A and B are statistically significantly better than more methods than C is in this count, right? This is one way of doing it, and let's say that makes sense, but I believe what I wanted to raise here is an awareness that ranks can be very misleading, and averaging over ranks is often going very wrong, so you have to be very, very careful with that. Okay. Standard error, performance, let's go into the performance for this entire log tree method, log tree 2, the 18. Overall it has an 18 state accuracy of 65%. Question? No? No. Now, on the first level decision, whether it's a soluble protein or one of these transmembrane proteins, the performance is 94 plus minus 2, so that's extremely accurate. And this is in fact immediately you see this is why that method does this step first, because it's the highest performance. And in fact it does this step almost as good or even better, as we later found out, as a method that is optimized for that step. So in our particular case, we put this still in the entire framework here, while the polyphobius is a much more complex method that uses much more than a single SVM to make that decision. So it's a remarkably accurate precision. And then we sort of show here that some decisions are done at fairly low levels of accuracy, and as you see here, so that's the standard error, 48. So we're getting into the realm where we just have so little data that we essentially, this is like the example of small data sets that I talked about the other day. For plasma membrane here we have 50 error, so the error is plus minus 50, one standard error. So essentially you cannot estimate anything, but ultimately that's the point we want to make. The numerical number is 50% with a standard error of plus minus 50. Okay, this is as good as we can get. We have this SVM. It does some classification, and there is some sort of limit. The accuracy is in red, the coverage in green underneath. Okay,
Quick question.
yes.
How did you determine the structure of the tree?
So again, this was done in this particular case. We tried to repeat what we did before, and we also tried all possible solutions, and both sort of corresponded to each other. There was a solution that was better than this, but since it did not make biological sense and was not much better, somebody in the group decided to keep this one. But it was better so biologically sense. So there are reasons you could argue you wouldn't on the top have the membrane decision, but you would have the membrane decision at each level. So you would first have the biological tree, so to speak, this one here, and then at every single step do membrane, not membrane. And that part we did because of performance. So there are some aspects here that are performance, and some aspects sort of a mixed bag. When we compared it for different methods, where we now do not have the same number of states here for eukaryotes, we compared to some methods for 5 states, to some methods for 9 states. Bacteria is on 5 states, and here this is for 3 states. And you essentially see that we mostly have better numerical numbers, and in many cases we cannot say that we are better because of the standard error that you see. So 65 is higher than 62, but given the standard error, we cannot establish at all that this is statistically significant. Statistically significant. So then you can still sort of argue, okay, we do more states. We have a numerically better number, and we can go 18 states, this can only go 9 states. So we beat that method, we at least do not do worse than that method on its own turf. But whatever, these are different ways of looking at it. Here's another way of looking at it, where we wanted to sort of somehow simulate mistakes in sequencing. So we remove the first 30 residues, we remove the last 30 residues, and we randomly remove a third of the protein somewhere in the middle, somewhere. And then we see that the method that we have, so overall it's 62, and for removing a random third we go down to 53. When we remove the beginning of the protein, we sort of have the major drop, because ultimately there are many proteins that have a single peptide or have zip codes at the beginning, and since we lose them by removing this beginning, that is why this drop is somehow so significant. Now you see that other methods that sort of perform somehow on a similar, and this is a different data set, that's the one where I said before the plus minus 14, because it was a much smaller data set of proteins that had not been used by any of the methods. Here we have some overlap, so they have used some of the methods for training in this particular case here that we used for testing. So they were not cross-validated, but it's the same data set at least. So here the difference is slightly higher. But you see that other methods are much more sensitive to removing parts of sequences. And again, most of these other methods, Wolf-P sort often uses the internal signal or uses amino acid composition. This uses essentially the k-mers. So most methods somehow use amino acid composition in a way that requires the whole thing to be there. Since we have the k-mers, the profile kernel allows you to make this, essentially it makes decisions. What we can see is the decision in which subcell localization you are is done on thousands of k-mers. Thousands of conserved k-mers, I should say. And some are missing, so that's why the accuracy drops. Your precision goes down when you lose some in between. And some are most important because they are very relevant, because they're at the beginning. They're very informative k-mers that you lose. But you still can make a decision. You can still essentially hold or keep up the accuracy closer to your performance for the full length than other methods do it. So this is another success. Now, the final step was to combine this de novo prediction with homology-based inference, and the way to do it ultimately is we looked how well homology-based inference performs, and then decided on some threshold. And essentially the simple algorithm is whenever we have a database of proteins for which we experimentally know the subcellular localization, there comes a new query, we run it against the database, we pick up anything at this e-value, we say that's the subcellular localization. If we cannot pick up anything, then we run the method that we showed before. Okay, that's as simple as it is, and it sort of does better than both alone. That is essentially what it says. That's the output where the predicted localization of this image here is in green, and where on the tree you see highlighted how the path has been chosen and what the reliability for each of these choices was in the final decision. One more thing. In this particular case, I'm going to just show one slide here. This is localization for entire organisms here, and what you see is differences between observed is the outer perimeter, predicted is the outer perimeter, and the inner perimeter is observed. And these observed, so here we have two pies that are based on different data sets. For all of these organisms, the experimental data is incomplete. So I simply take whatever I have as experimental data and use it to infer or assume that everything that I don't have looks essentially in the distribution like what I have. And what you do see is that the inner circle and the outer circle, they, for all of these organisms, look very different. Even for yeast, for yeast they are most similar in some sense. No, not in some sense. They seem to be most similar because here is the highest fraction of experimental data that we have. But for all the others, they look very different. And when we look in more detail, there's a lot of evidence that the prediction is the right one. So yes, the prediction makes mistakes, but since it can predict for the entire organism, the estimate for the entire organism is more accurate than for the more accurate experiments that are completely incomplete and are very biased for what you can do experiments. For instance, secreted proteins are difficult to do experimentally still. For predictions, they are as good as everything else. In fact, they are better. The prediction of secreted proteins, and that is why what you predict here is actually better than what you observe.